This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 977, recorded on January 20th, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here at the incubator in New York City, Dixon Despommier.
Hello there, Vincent. And the weather outside is, who the hell knows? You don't have any windows.
It's a studio. You can't have windows.
And who cares? We're totally insulated from the outside.
I got a text from Amy. She wants to know if you showed up.
If I showed up?
Yeah. I guess she has faith in you, right?
Who did I show up?
Dixon, it's 8 degrees Celsius and mostly cloudy.
Yeah, that's about right.
Here in New York City. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. It's basically the same here.
From Western Massachusetts, Alan Dove.
Good to be here. And the weather outside is, it's 36 Fahrenheit, 2 Celsius, light rain, fog, mist, occasional snow.
Wow.
Fortunately, none of it's sticking to the roads so far.
And from Montreal, Canada, Angela Mingarelli. Welcome.
Hello. Thank you for having me again.
No, we're not having you. You're on the you're a host.
True. I know. Here, I think I have you all beat. Well, mine is closest to Alan's weather. So it's the same, but it's 30 degrees. So -2. I think it's like, or 28, I think, in Fahrenheit.
Yes.
And it's snowing.
Wow.
It snowed a lot, actually. Last night it snowed like 15 centimeters.
Oh, my.
And it's been snowing all day.
Cool.
Does it get cleared quickly?
Yes, it does. Actually, Montreal is very efficient with, well, they could be better, but compared to other cities, it gets cleared, I guess, fast enough. If it's always snowing, it's hard to clear the sidewalks.
As long as it's not like Buffalo, you're okay.
No, a little bit less.
Canada knows a thing or two about snow.
I'm sure they do.
All right, we have a couple of announcements, and then we have 2 papers for you today. First of all, remember Amy Rosenfeld at the FDA has a research assistant position that she's recruiting for. So we'll have a link in the show notes. It's basically for a research assistant to help with her work on enteroviruses, like Entero D68 poliovirus, human rhinoviruses, genetic, biochemical, cell-based studies for understanding immune responses to infections and also developing small animal models. All the work is carried out under BSL-2 conditions. So there will be a PDF where you can find Amy's email and you can ask her for more information or just apply for the position. And also the American Society for Virology, the annual meeting in Athens, Georgia, June 24th through 28th. Abstracts are due February 1st. That's
10 days from now.
Thank you very much. I'm not good at math. You know that.
I had to take my shoes off for that one.
Boy. Travel awards for global scholars, which require an abstract, are due January 23. That's the day after this episode drops.
And global scholars means not in the U.S., right?
Mm-hmm.
It doesn't mean you got your PhD in geography.
No. Right. And then other travel awards, trainees, teachers of undergraduate virology are due February 3rd. Some of these require ASV membership, so sign up for that now while you are working on your abstract. ASV.org/ASV2023 for information.
I just wrote it down.
You're going to do that, right? You're going to work on
Exactly, yes.
And finally, Microbe TV has a Discord server. Now, there's some of you who will know what that is. It's great. We'll put a link to sign up for that. Those of you who don't know what it is, Discord is a community where you go and you do text chatting. It's the largest text chatting community, and you can talk about all kinds of things. There are separate servers for different subjects. There's gaming, there's needlepoint servers. I don't actually know if there's a needlepoint, but
I'm sure there's a needlepoint. There's a server for everything.
I'm sure there is.
I was already on Discord for some programming and game development stuff, and so when I saw this, I went ahead and joined.
Angela, you know what Discord is?
No, I actually haven't used it. Like I've heard of it, but I don't actively participate.
I need to sign up. This will be my second Discord server that I'll be a part of.
What's the first?
Jeopardy alumni.
That's good.
There's some discord there, by the way.
That's good. Yeah, so this is Microbe TV, which is basically a place to talk about everything covered by our podcasts, life sciences. And so it's a good community. I have an admin and a moderator, a couple of moderators who they have set up the server. They've put various rooms for different subjects. So that's very nice. And channels, they're called. Okay. And there are over 100 people there already. So check it out. There'll be a link in the show notes. We'd love to have you join. You can ask questions. You can converse, any time of day that you want. It's open 24/7. It's cool. I actually opened it a year ago, but I never did anything with it until a few people volunteered to help, and that was the key. So you should check that out. That's a lot of fun. All right, we have a snippet and a paper for you today. And the snippet is a Nature article, SARS-CoV-2 infection and persistence in the human body and brain at autopsy. This is by a large collection of people all over the place. Looks like a lot of people at the NIH and other places as well. And, so the question is, does SARS-CoV-2 persist more than the 7 to 10 days than the acute infection? So acute virus.
Just to do authors real quick, Sidney Stein is the first author. Daniel Chertow is the last author. And there's the gazillion people listed, and also the NIH COVID-19 autopsy consortium, which I assume is a gazillion more people. So this was a very large collaborative effort.
And this has, one of the authors is Emmy DeWitt, who was on TWiV. She's at the Rocky Mountain Laboratories. I recognize that name. Vincent Munster, also there. He was also on TWiV several times. All right. When there are a lot of authors, I get, I just don't want to do it. Thank you.
Yeah. No. We're not going to read all the authors. That's
So does the virus persist longer than the acute infection of 7 to 10 days or so? And so what they have done here is to carry out autopsies on 44 COVID-19 cases. These are all unvaccinated people. So it's April 2020 to March 2021. All unvaccinated individuals who died with COVID. So notice how they wrote that, with COVID. There's a big debate going on now, whether you die with or from, right?
Yes.
Are you admitted to a hospital with or for COVID? It's a very hard question to address, because when you go to a hospital now, you have to have a COVID test. And
I am pleased to say, hospitals are starting to phase out the routine testing.
Yes, they are.
Because this was early in the pandemic. It was the critical thing that we needed most. If you go back to our episodes then, it was, oh my gosh, why can't we do more testing? Because that's what we needed to try and contain spread. And now that this virus is endemic, testing for it is just generating noise. And you're getting, anybody who's admitted to a hospital will get a rapid antigen test, and it comes up positive. And well, that's not why their leg is broken, right? That's they're in the hospital for something else.
But that goes down as a COVID admission, though.
Yes.
In many places. And that's right.
True.
If you look at one of the extended data figures for the paper, it's extended data figure 2. They have a table with what was listed as the cause of death for all of the patients.
Yes.
They only have COVID listed for one of them, but almost all of them were either sepsis or pneumonia or diffuse alveolar damage. And so I took that at least to mean that largely these patients were suffering from COVID infection and sequelae thereof. Yeah. They mentioned in one place that there was someone who had a seizure that they thought was unrelated to COVID. But from what I could tell, these people were all positive, and it seemed as though the disease that they died from was some sort of COVID-related pathology.
Yeah, and this is early in the pandemic when that was why people were going to the hospital.
All right.
What were
Anyway.
the dates of this specifically? I didn't. This is
The
2021.
patients were April 2020 to March 2021.
Yeah. Okay.
So that's
Right.
why they're all unvaccinated.
So then they, then they looked for viral genomes by PCR. It's a very well done study, I think. They did things very extensively and very carefully, and they do PCR. They look at virus, infectious virus. We'll talk about that. They also do genome sequencing. They do in situ hybridization. Right. So by PCR, they find they can see viral RNA in 84 distinct anatomical locations. Of course, the highest RNA level is in the respiratory tract, always, right? Because that's the main place where this is reproducing. They say, and this is a key point of the paper, they say the RNA is detected across multiple tissue groups among all late cases, so the ones that died longer after infection, despite being undetectable in plasma in any. It's also found in the central nervous system in 5 of 6 late cases, including one patient who died at day 230 after onset. So they still find viral RNA there. Now,
another important point I just want to bring up here is they standardize their procedures to the extent possible for these autopsies. So one of the issues when you're looking at postmortem data is, well, okay, how long did the body sit around? How was the autopsy done? Which tissues did you section and which didn't you? But in this case, because it's the NIH autopsy consortium, I guess they were able to come up with a standard procedure, and so these samples are actually comparable.
When they talk about the early versus mid versus late patients, that's based on something relative to the day of kind of illness onset.
Yeah.
But in every case, these patients are still ill.
right.
Yes.
Right. So previously, other groups have detected viral RNA in non-respiratory tissues, and they always say, well, a little bit gets in the blood and it spreads to other tissues. Doesn't mean it's actually reproducing there. And so they say, well, we don't find it in plasma, so that can't be an explanation for what we see. And I can't really argue with that. I mean, I would like to argue, but I can't argue. They don't find it.
Well, they also, we're going to get into. They go further than just PCR as well.
They also that those results are for genomic RNA. Then they look for subgenomic RNA, messenger RNAs, and they also find this as well. And that's more suggestive of reproduction, actual virus reproduction, because the genome can just be there, but if it's being copied into mRNAs, it may be reproducing. More likely to be, I would say. So they find that those results are consistent with the genomic PCR as well. And then what they do is they take some of this tissue, which is ground up, and they put it onto viro cells in culture, right? And they say, do we isolate virus? And they can isolate virus from 25 out of 55 specimens, okay, over a wide range of CT values or CQ values. But it's not quantified. This is one of my complaints about the paper, that I don't know how much virus is there, because if you put a piece of tissue in cells and there are a few PFU, it'll score as positive. And I don't know what a few PFU would mean in terms of pathogenesis. I would have loved to see a quantification of the tissue, but I understand that that's difficult. We used to do that all the time with mice, right? We'd infect them with polio. We'd grind up the brains and do plaque assays. So I'm used to it, but not so
you can't
easy.
just grind up the whole human brain into a plaque assay. That's a little cumbersome.
Yeah.
So that's, I think, my main beef with this, because as you see, they're going to conclude that we prove that there's extensive extra respiratory reproduction of the virus, and I'm not sure we can say that, but...
Do you think the tissues were too autolyzed for EM work?
Did they do some? I think they did some EMs later on, if I remember.
Because if they were finding whole particles, then that's signs that it's reproducing.
Does anyone remember?
I only remember the in situ and sort of the histo
no,
path.
I'm thinking of the other paper.
Yeah.
Yeah, the other paper.
There's
The
lots
other paper.
of
So
EM
no
there.
EM. I don't know. Maybe it was, not suitable
far
for.
gone.
I just don't know.
Yeah.
But if I saw one particle, I still wouldn't say
No.
right extensive reproduction. They did some viral genome sequencing, and they see some synonymous substitutions. In other words, changes that still code for the same amino acid outside of the respiratory tract, which they say is additional evidence that there's reproduction, right? Because it's different from the virus in the respiratory tract. Yeah. Okay. And then they do in situ hybridization, where they're looking for, in this case, spike mRNA in tissues. You take a tissue section and you hybridize it with a nucleic acid probe, which will show you the presence of viral nucleic acid inside of cells. And they have a very nice image showing this, where, the RNA will show up as a color, and you can see it's inside cells, right? So I think they said, how many of these? 35 different cell types. How many people did they do this in situ with? And I don't see the number popping. It's 16 cases covering a 4-log range of N gene copies. So they do see this, and the pictures are pretty. You can see the cells, nice morphology, and you can see viral RNA in them. I don't know if that means there's active replication or not.
What would active replication look like?
Well, one of the things they note is that there is no cell lysis or inflammation in any of these sites.
Yeah, but this is dead tissue, though. Come on.
Well, it's dead tissue now, but it was recently alive. And
it's a good point.
If the virus was in there replicating, and this is a lytic virus, you might expect to see some cell lysis.
Yeah. Brianne, would you expect inflammation to go away immediately after death?
I don't think that you would. No, you can certainly see inflammation in other tissues in other disease states by autopsy. I wondered more about, whether or not, for obvious reasons, I was thinking about, what's going on with sensors in those cells to be sensing viruses and things like that. And I didn't end up taking a look at some of the data on that I had planned to look at. But you certainly could see inflammation.
Yeah. So that may suggest that maybe there's RNA around, maybe there's a little bit of infectious virus, but it may not be reproducing extensively. I mean, we know now for RNA viruses that they can persist for a long period of time. Diane Griffin was one of the first people to show this, that we think these are viruses that come and go, but actually the RNA sticks around a long time.
Yeah, and I also think that these figures are particularly important because of that idea that had been out there previously that this is only because of blood being in some of those tissues that the virus happens to be there. We can see that it's actually in the cell types of the tissue.
Yeah.
So
I guess it's kind of odd that.
Go ahead.
Sorry.
No,
that we see this like in figure 2. You can see that it's obviously inside of like many of these cells, extrapulmonary tissues, and that there isn't any inflammation is kind of odd, I find.
Yeah, that's why I'm sort of wondering, particularly when I'm thinking about the brain, how much sensing really there is, and how much we would see inflammation in other cases as well. I think it may be kind of a immune privilege dampening of not having a lot of inflammation in some cases in those tissues.
What does the word exemplar mean? The best example of?
A really good example.
A good example of.
An example that you hold up.
Okay, so this
an example that exemplifies.
So they say these are exemplars. So maybe these are the best sections they saw. Maybe they're just a few in every tissue. Spotty patches of infected cells, and that's it.
And they're really diverse tissues too. They have spleen, adrenal gland, endometrium, testes, esophagus. Like they're very, very diverse tissues.
Yeah.
And the fact that we shouldn't say replication, I guess, but we can see RNA inside of the cells and that there's no inflammation whatsoever, at least in these. Maybe there was in other sections, I wonder.
So they're still looking for a cause of death, is that right?
The cause of death of these people has already been stated on the death certificate, right?
Yeah.
And as
They
Brianne
didn't
said,
die
these
for
are
a cytokine storm or anything like
right.
that,
So these are
obviously.
death. These are causes of death that I think, with only one exception, are completely consistent with dying of COVID-19.
Yes.
It's pneumonia. It's the usual things. And there's one kid who had a genetic
A
seizure
seizure.
condition and had a seizure and died, and that's okay. So that kid died probably of the seizure, but everybody else seems to have died of COVID-19 or related complications.
I had to look up, they listed a lot of the causes of death as DAD. And so I had to look up what DAD was as a cause of death. It was diffuse alveolar damage.
Mm-hmm.
So anoxia, you think they died from low oxygen levels in the body?
Well, yeah, if the
Perhaps.
I mean, you have to nail it down to some physiological event.
Right. DAD is listed for many of them.
Okay.
Mm-hmm.
Okay.
Dad killed him.
Right.
So that's pretty much it. They also see RNA in the brain, but again, they see no histopathology anywhere. So they say these findings conclusively demonstrate that the virus is capable of infecting and replicating within many tissues, including the brain. And it just brings me to mind, we had a neurologist from Columbia on 2 years ago, and they had done a similar study at autopsy and found no evidence for
Right.
virus. Jim Goldman was
Yeah,
involved
yeah,
in
yeah.
that study,
Remember.
and I forgot her name. She's escaping me, but
Kieran,
Thacker. Thank you, Kieran
Woof.
Thacker. She was the neurologist who was on that paper, and she came in and did the TWiV. So she said, I remember asking her, is there any evidence for reproduction? She said, no, we don't have any. So I don't know why there would be a difference in the two studies, but obviously we need more work to sort it out.
Well, the
Now-
closer you look, the more you see. So no
suppose.
evidence for viral reproduction
Yeah.
means you didn't see it. And these folks
Yes.
saw it. They also did a very, very detailed search for this exact thing. So
But,
it may just be, it was there, but it wasn't visible with the available assays
So I
or
think-
the assays that were used.
yeah, there's some infectious virus present, but it may be low. We don't have a quantification. And there's no inflammation. There's no cell damage. There's no histopathology. So I don't know what relevance to the disease it has. Okay. Because I know these individuals would like to think this has something to do with long COVID, right?
Sure.
But I don't see any evidence that it's contributing to extra respiratory symptomatology. I mean, I agree that in the nasal epithelium, there's evidence for, destruction of the support cells and also lymphocyte infiltration that may be persistent. That's fine. But here, I don't see convincing evidence that any of this would be playing a role in disease, I guess.
A really difficult, expensive study that I would love to see, that would be hard to get done, I think, is to do this for some other RNA respiratory virus. Do it for,
Yes.
pick a maybe rhinovirus, maybe whatever.
Influenza, where people
Influenza,
die, right?
sure.
Yeah.
Actually, flu would be a great choice because you could get a bunch of people who definitely died of flu or of flu-related complications.
Right.
And go search as deeply as this with these same procedures and see if you find flu, because long flu is not really a thing as far as we know, but if you get similar results, maybe this is just something that RNA respiratory viruses do.
Right. And I
Yes,
think the
yes.
other piece, and again, this would be such a hard study to do. I don't know if you could totally do it, would be, these patients all died of COVID. Even the ones who were sort of long or the late time points were really dying of COVID related pathology. And so what you'd want to know if you were sort of tying this to long COVID is, can you still see this after the patients have recovered?
Yeah.
And it's not clear that any of these patients had recovered.
Right.
And we could do the same thing in Alan's cohort as well, just making an even more impossible experiment.
Yes.
Yeah.
I wonder if, because
I
they
like
don't know.
that. Go ahead. Sorry.
Sorry, they don't know. I guess it doesn't say if this was they didn't sequence the actual virus, did they? Because we don't know if it's the
Yeah.
ancestral strain or if it was one of
Yeah,
the
they did.
variants. Or if,
They did sequence.
They did genome
so is this?
sequence.
But I mean, they did genome sequencing, but for every single patient.
No, for
So do we know?
a subset. Yeah.
Mm-hmm.
1,
Because I
2,
wonder.
and 3. It looks like they did. Wait, we used genome sequencing to look at spike gene only in a total of 46 tissues in 6 individuals. Just the spike, though, not the whole genome. Yeah.
Yeah, I wonder if depending on the variant, because obviously at that point in time, I'm not sure which variants would have been circulating already, if that would influence the tropism of the virus itself.
Yeah, they actually say that this was before the emergence of variants, so we don't know how that would affect the tropism. Yeah, I do think that they say that these are all from old people who were unvaccinated and died.
Well, except
And-
for the kid.
Except for the kid, they had pre-existing medical conditions. So who knows whether the same would happen in a young, healthy, or a vaccinated person, right? They may not.
And with the variants, yeah, the things have moved on since this, so we don't know that the current infections look like this.
And they say, although it is tempting to attribute clinical findings observed in PASC to viral persistence, our study was not designed to address this question. Yet you bring it up.
Because it was tempting.
It was tempting.
Well, and you also know that the readers/press might bring it up.
It has not escaped our notice that
Yes.
this could, yes,
James Watson. and
this is a Nature
Francis
paper.
Crick. Yeah, I mean, obviously that's in the back of everyone's mind, but I'm not convinced that because of all these limitations that this has broad. And I think looking at another virus would be great because I suspect you'd find it with other
Yeah.
respiratory viruses as well.
A lot
I
of the
wonder
stuff
with
that we're
the-
seeing with SARS-CoV-2 is just because I think this is the first virus we've looked this deeply at things like this.
Yeah.
Go ahead, Angela.
No, I was just thinking about the human endemic coronaviruses as well. That if they would,
Yes.
if possibly we have 229E, sometimes post-infection that's also in the, I don't know, the spleen, or if it's in different organs, or NL63, or all of these other coronaviruses that are
OC43,
less
yeah,
pathogenic.
that
Exactly.
might actually even be a better choice than flu to look at for
Just
comparison
that
to this.
you don't die of those, so.
Well, do you?
some, well, most people don't, true.
Well,
but-
actually, you could take just random autopsies and look
Yep.
for those genomes. Why not, right?
Yeah.
That would be very few, though, because they don't do a lot of autopsies nowadays.
No?
No.
They do not.
Why is that?
I think people refuse them for various reasons. And
Well, and if somebody is very old and dies and was expected to die sometime soon, and okay, they're very old and they died and that was the expected outcome. And
that's
Yeah.
right.
I mean, you're not gonna get a lot of hospice patients that are gonna get autopsies because there's not a good reason to.
Yeah. And so if a younger person dies unexpectedly, then you'd have an autopsy?
Perhaps. Whenever
Yeah.
the death is suspicious, they do it. But for most routine expected deaths from long-term illnesses, they don't. And
Yeah.
Columbia had a hell of a time training residents in pathology due to that. They didn't have enough autopsies to really do it.
I remember Kieran said that in that work, which I mentioned, they actually got permission from the patients. They knew they were going to die, and
Oh,
they
yeah,
went to
that's
them
right.
and they said,
Yeah.
"Can we do an autopsy on you?"
That's right. That's right.
Which is tough, right? It's a tough
Yeah.
one.
Yeah,
I would say, yeah, sure, go for it.
Yeah.
that's right.
for sure.
But not everybody would. All right. Anything else? Is that good enough?
I think that's good.
I think that's a snippet.
Is that? This is
30
doesn't
minutes
tell,
into the show, we're finishing up our snippet. Yes.
doesn't tell us much about the pathogenesis, right?
Well, no, it's just showing, I think, that there's SARS-CoV-2 makes its way into other tissues.
Yeah.
Does it do anything? We don't know, but we
Right.
know it's there. I guess it's still an interesting finding to
That's
know
a
that
good.
it's.
It's a good summary. Yeah, in
in
old
other
people, mostly
organs.
old people who have comorbidities and who died and were not vaccinated. All those.
It'd be
kept-
interesting to see young people. If I had an acute infection,
Yeah.
obviously you're not going to take a biopsy of my spleen. I mean, if I died from another cause, and
Yeah,
then,
then you
could
could.
look.
Yeah.
But this is obviously
All right.
very difficult.
All right, on to our paper. This was selected by Angela. So Angela will have the honor of leading our discussion.
Yes, I did. So actually, Dakota Rogers in my lab, one of the other PhD students, he first mentioned it to me. And I saw it and I thought it was really interesting because the findings are super interesting, but it's definitely out of my immunology scope. So I will need your help in interpreting the paper. But yeah, so the paper is called, it's a paper in Cell and Cell Press, and it is Resurrection of Endogenous Retroviruses During Aging Reinforces Senescence is the title. And the first author is, I'm sorry if I mispronounce these names, Xiaoqian Liu. And there are 3 corresponding authors, I believe, Weiqi Zhang, Jing Ku, and the last author is Guanghui Liu. And they are from mainly, let me see, the State Key Laboratory of Stem Cell and Reproductive Biology, Institute of Zoology, Chinese Academy of Sciences in Beijing, China. And this group, the Guanghui Liu's group, does a lot of aging and regenerative medicine. I think he had like 283 publications when I looked him up on Google Scholar and ResearchGate. Very impressive. And
I think there might be 4 co-first authors.
Yes, there are 4.
Oh, 4, sorry.
So in addition to Xiaoqian Liu, there's Zhenpeng Liu, Zheming Wu, and Ji Ren are all first authors. And I also apologize if I butchered the names.
Oh, sorry. Yes, you're very correct. I'm sorry. Yes. Sorry to them if they listen. And giving them all the credit. And yeah, this group does a lot of, from what I saw, I looked through some of the other publications, a lot of single cell sequencing and aging, and they have multi-species comparisons, and they seem like a very established lab, especially in aging and in cellular senescence. So yeah, this paper is extremely interesting. So it basically, which a lot of people don't know, humans, not only humans, but other primates and other mammals, we have these retroviruses that have integrated into our genome over thousands of years that millions of years, that comprise up to 8% of our genome. So to think that almost 10% of us or 8% of us are retroviruses is kind of cool and kind of horrifying at the same time. And yeah, most of these retroviruses, so they're called, I think in the paper they call them ERV, so endogenous retroviruses. They are mostly, what's the word? Not inhibited, but latent. They're non-replicative, and
I guess they don't make infectious particles, right?
Yeah, I would think of it as inhibited.
Mm-hmm. Yeah.
Right, and a lot of these, the older ones, and you can do genetic clocks to figure out approximately when this came into the lineage, and a lot of the older ones have accumulated a lot of mutations that render them effectively non-functional.
Mm-hmm. Yeah, so one of these viruses, a recently integrated virus, so some of them, like Alan said, have been in a part of our genome for millions of years, but the human mouse mammary tumor-like virus 2, HML2, which is called, I guess, HERVK is how is the acronym they use in the paper, maintains some open reading frames and open reading frame encoding proteins that are required for actual viral particle formation. And they see some of these are GAG, POL, MPRO, etc. And they see that some of these in this virus are actually open still. So there's the potential for replication of, I guess, virus in this case, or viral-like particles.
Yeah.
Exactly, production of particles, compared to some of these other endogenous retroviruses that don't have this capability. And yeah, so basically they
So
first
the idea
start
is
out.
to say, is there a relationship between senescence and these retrovirus particles, right? That's the question.
Sorry, yes. So basically they say, do these endogenous retroviruses or the potential replication of these viral-like particles, could these be contributing to cellular senescence in any way? And they'll have, so there are different progeroid syndromes, so different like aging or senescence syndromes like, what is the name? The Hutchinson-Gilford progeria syndrome and Werner syndrome, as they use in this paper as two of like the senescent cell models. And they can see that, so in figure 1, they're basically showing this epigenetic de-repression, as they call it, how these endogenous retroviruses have been repressed, and then this de-repression of this HERVK is observed in, as they call HMPCs, so human mesenchymal progenitor cells, which are the cells that they use in culture. So they take these cells and then they look at either late stage and early stage passage cells, and then they look at the different phenotypes. They see that there are increased LTRs in these late stage passage cells specifically. What else do they see? They see the HERVK is increased in senescent cells. So when they look at the progerioid phenotype, they can see that the HEVRK specifically is enriched in those cells. And some of the retroviral elements of senescence, they measured via qPCR, like the GAG, POL, and EMV, which are some of these necessary elements for viral particle formation, are also increased in the late passage cells in culture.
So one thing that they don't ever flat out say, or maybe they say, but they didn't talk about a ton. And since I'm not a senescence researcher, it wasn't something that, I necessarily knew. I kind of had to connect the dots myself a little bit. Is I think the idea is that in these cells that are aged, or whether they're either late passage or from these syndromes, there's a general de-repression. And so there's a general de-repression. DNA is chromatin's less methylated, so we don't have a lot of the epigenetic repression. And maybe they previously said that's part of the problem of aging. And now they're trying to tie this to, maybe it's the de-repression of these specific genes. Mm-hmm. It's not just across the board de-repression. We think it's these genes, and as Angela points out, the particle formation. And it took me a while to get that piece in all of this.
There's a lot in this paper.
Yeah. There's a lot.
Which is open access if people want to dig into it.
Yeah, there's a lot. When I said that I would do this paper and then started going more in depth into it, I was like,
What did I get myself into?
But it's really interesting. It's really interesting, though. Like the findings themselves, I think are I don't know what some of this controversy Brianne was talking about on Twitter, but
so I can actually it's actually related to figure 1, and I can say what it is, and that's about as much as I can say about it. Apparently, some people who work on ERVs think that some of the RNA-seq methods that were used here were not the right RNA-seq methods to use for endogenous retrovirus sequences, and so they're not clear that this actually shows the transcription of some of the things the authors say because of the RNA-seq method used. And that is the sum total of my understanding.
Okay. Well, this is, fortunately, they have more than RNA-seq data in this paper. Right. Mm-hmm. And it all actually ties together quite nicely, I think, right?
Yeah, so then they also do, let's see, where are we? They also do... Sorry?
Proteins and particles. Is that where you wanted the next section, right?
Then they see in RNA fish, they can also see that this HEVRK, they can basically see a transcriptional region, regulatory region of the HEVRK in the senescence HMPCs via fish in E, which is, I guess, nice to see. And then...
So this is in situ hybridization for fish is fluorescence in situ hybridization. Yeah. It's a technique
like what we talked about in the previous paper.
Yes, just like in the previous paper, they're looking at expression.
Exactly. And then I guess that's figure 1 is more or less that. They also show that there's actually I think this is important. They show that there's also reduced CpG DNA methylation, as Brianne was saying, in specific regions. So it was the HEVRK int regions and proviral loci, and then also there were decrease in repressive histone marks like H3K9, ME3, and an increase in transcriptionally active histone marks as well. The names, I guess, are unimportant, but that's also interesting to see that these data indicate, as Brianne said already, there's this epigenetic de-repression of HEVRK that could be contributing to the actual transcription of the virus, so HERVK. And they could be associated with cellular senescence.
Right. So HERV-K is normally bundled up with the inactive
HERV-K.
genes in these sort of storage nodes, and then things are coming unglued here, and HERV-K is being expressed.
I like that.
I think
Unglued.
HERV-K.
Unglued, yes.
Yeah.
The center cannot hold.
Yeah. So then in figure 2, they have this, they actually show the accumulation of viral proteins and these RVLPs, so retroviral-like particles of HERV. I'm going to use that, Alan, HERV-K. That's so
HERV-K,
much
yeah.
easier to
Yeah,
say.
so
usually is.
I do this with all of the endogenous retroviruses, because if you keep saying endogenous retrovirus, ERV or whatever, it just gets tiring. So they're all ERVs, and they're HERVs because they're in humans.
And Angela, do you know what they're called in pigs?
Pervs.
HERVs.
Yeah. Yep. I actually,
Yeah,
at the end of the paper, have a question about pervs.
HERVs,
So-
yes. Well, PERVs are big news because they're a big problem in xenotransplantation.
Right.
We could use pig
They are a
organs
big problem.
Yep.
Yes.
if it wasn't for the PERVs in the
Yep. Okay, sorry, I had to
Sorry.
tell you about that.
So yeah, next they were asking whether these elevated endogenous retrovirus expression could elicit this production of HERV-K protein components and then the formation of these viral-like particles. So in figure 2, we can see they do, in A, we can see an increase of HERV-K envelope. And I think this is, again, A, I think is also fish, isn't it? Yes.
Looks like it.
They don't say one. Yeah,
Yeah,
it says
immunofluorescence staining.
Yeah, immunofluorescence staining, where we can see there's also increased fluorescence intensity in the HGPS and the WS, which are the progeroid syndrome lines. I wish there was an easier way to say that, but
Yeah, I realized quickly as I was reading that I was like, I can't remember that word. They're the mutant cells.
Yeah.
the mutant cells, yeah, the
that's
old
it.
cells,
Yeah.
the
Old
old
cells.
cells. Old
elderly cells.
Hey.
Well,
hey.
but their progeroid is it's this genetic disease of
Yeah.
premature aging.
Yeah.
Exactly.
Benjamin Button in reverse.
Benjamin Button. I was just going to say, what was that
So
movie?
they're actually the elderly young.
Yes.
Yeah.
Yes.
The elderly.
That's true. Progeroid, not progeroid.
I don't know how to say it.
Progeria, I thought, is the
Progeria,
condition,
no?
right?
Well, it's progeroid. Progeroid.
Progeroid.
Yeah,
Okay.
progeroid or
Progeroid.
progeroid, whatever you want to put the accent.
Progeroid.
But it's
Progeroid.
progeroid. Don't put
Okay.
an extra I in.
Okay.
Cells from
Okay.
people with the syndrome.
Yes.
Yes, old cells. I said, okay,
cells.
that sounds, I guess, or old
You could call them.
cells.
You could call them button cells if you want.
button cells. So basically, they're just showing that these button cells, that these progeroid
Mutant cells,
have
yes.
mutant cells have increased. So HERV-K, ENV, and then also P16 and LAP2 that are all associated with cellular senescence. And what else did they? Yeah, they're showing also that in early passage cells in wild type, so I guess young cells, they would consider them young cells, we don't see this phenotype. And then, but in the mutant cells, we do. So in C, you can actually see, I think C, they're actually showing you, we can see the RVLP, so the retrovirus-like particles we can actually see in the cytoplasm
This is our electron micrograph.
of,
Yeah.
exactly, electron microscopy for Dixon. And we can actually see the retroviral particle formation, which I think is really cool. But you can't see these in the early passage younger cells. And yeah, these are anywhere from like 80 to 120 nanometers. And again, we can see them in D. They're just showing the same. They're showing in wild type versus the mutant cells. We can see this increase of the HERV-K RVLPs. And then they also did it, I think the next one is in fibroblasts. So then they take fibroblast human cells to verify in other cell types if they see this early passage versus late passage RVLPs. And yeah, right? Or am I? Yes, exactly. Exactly, yeah. So we can see the, in primary fibroblasts, they found HERV-K envelope, HERV-K ENV DNA. In late passage was the fluorescent intensity was much higher than that of the early passage cells. And then we can also see, what else did they do with fibroblasts?
Particles.
They were looking at the histone marks. Yeah, but apart from the RVLPs, I think they were looking at the histone marks as well. And they saw, let's see, an increase.
Decreased occupancy of these methylation sites, right?
Yeah, so it's that same hypomethylation.
Yep.
First, they'd seen it across the board in old cells, and now they're seeing it in the mutant cells. But they're looking now specifically at these sites instead of just across the genome.
Yep. You get increased expression in transcripts from old people compared with young people. Fivefold, yeah.
Exactly. Okay, and then then on to figure 3, which was some of these
And they
panels up to
don't
panel
identify who in
Q.
the authors list was the old people and who was the young people.
You think that's where they came from?
No, I don't know where they got these, but just
Yeah, they might have. Yeah,
they might have.
some cheek cells. Yeah,
Yeah, the tenured and the graduate student cells.
The first year and the 6th year,
yeah.
the PhD students.
Yes,
I'm sure my HERV-K is going to be
heavily
way higher
aged
by the end.
cells. Yes.
I don't know. It depends how long you plan to be there, right?
Not too long, but
Yeah.
even so, even after 2 years, I'm sure they've increased exponentially. So then in figure 3, they to determine how the activation of this endogenous retrovirus affects cellular senescence, they used a CRISPR-Cas9 mediated transcriptional activation system that contained activation protein complexes with sgRNAs that were targeting this HERV-K LTR5 HS promoter. And in the wild type HMPCs. So what they see here in figure 3 is, so there's just, they're confirming, and then they see this beta galactose, I believe is part 1 of the SASP protein. So I didn't say that, but the secretory associated senescence products, or is it proteins?
I think it's proteins, but I don't remember.
SASP, which are associated with cellular senescence in general, and one of them is specifically this beta galactose?
It's galactosidase.
Beta-galactosidase. Beta-galactosidase. So they see an increase in this beta-galactosidase in the HERV-K post the, let me see.
The ones where they turned on the promoter, basically.
In B, yeah, exactly. They turn on the promoter and they do see an increase in this marker of cellular senescence. And then they also see a decrease in Ki67, which is a proliferation marker. So the opposite. So when they turn this on, they see that the cells proliferate less. So obviously also showing this senescence phenotype. And then there's so many panels in
Yes.
Let's see, what else do they show? In 3, I'm trying to get to the most important things in 3, I would say.
Yeah, it goes up
also
to
showed-
3Q. So they're yeah.
It's the Q, it's so much.
I know. I was really glad they had this figure
Mm-hmm.
because I feel like as I was reading, figure 1, I was like, okay, so you're telling me that there's de-repression in senescent cells. Does that mean that if you're senescent, you de-repress, or if you de-repress, you become senescent?
Right.
And here they have this nice figure in all the cell types and
Yeah.
in all the different, ways of looking at it to show, no, if we actually purposefully de-repress, the cells start to look senescent.
Right.
This is the
True.
causality shot.
Yeah.
Yeah, and they do all the markers that they've looked at, right? They're looking at chromatin methylation, they're looking at transcription, they're looking at senescence markers. Yeah, it's a good
DNA
experiment.
methylation. Yeah.
And I also was happy because I was like, oh, that means I'm understanding the paper if this is the next figure I was looking for.
Yes.
And they also
Yeah.
do they use 5-AZA to demethylate
Yeah.
chromatin, and that upregulates the HERV RNAs, right? And it leads to senescence as well. They do knockdown, which does the opposite. They also do some CRISPR to knock out these elements. It's quite nice. Makes the link
Yeah.
between the endogenous, the HERV-K, and senescence, I think, pretty good.
Yeah.
Exactly. And also to say that with this figure and the many, many things they did, and I'm sure it took them a very long time to
Yes.
show that these
And a lot of
enhanced
co-first
levels,
authors.
Yes. A lot of people working. I'm sure this took a long time and it's a wonderful study. But yeah, they revealed that there are these enhanced levels of endogenous HERV-K that are driving this HMPC senescence. Which is actually so interesting when you think about it, that this, like literally that 8% of our genome are these endogenous retroviruses, and that one of them that was integrated, we say recently, but I'm sure instead of millions of years, it was like hundreds of thousands of years.
I think it may actually be a few million. I was looking up. I didn't find.
Oh yeah, they're not thousands. No, it's
Yeah,
millions.
they're like these have been with us as long as we've been a species.
this one
Pre-Homo
specifically-
sapiens for sure.
This virus, though, they say was more
This
recently
one,
integrated.
this one is
We
more
just don't
recent
know.
than some of the other HERVs that are like since we had backbones. But
Like notochord, since the
Yeah.
notochord.
But it's still pre-Homo sapiens, right?
I
I
think
believe so. Right, because don't they look at it in later things in primates?
Yeah, we're getting to other great apes.
Homo
shortly,
sapiens
right?
is only 400,000 years. So
Yeah, we're
Yeah.
young.
Oh, wow. Yeah. Well, they say exactly. They say that we got them from primates previously from other
Yeah.
primates.
So
and they-
HERV-Ks began to appear in primates 30 to 35, in old world primates, 30 to 35 million years ago. But those are the broader family, not the HERV-K.
Not this
The
specific.
HERVs, let's see if we can get, HERV-K is an extinct blah, blah, blah. Getting a number
Yeah, I
here.
was having a hard time getting a fix on exactly this virus when we think it came in.
So HERV-K has been resuscitated by two different groups because you can correct the mutations and get infectious viruses.
Right.
And I think that's because there aren't so many changes, right? Because it's not been
Yeah.
around that long. Because if it were 30 million years, it would be really hard to fix it. But I can't find quickly how old it is. But if you want to keep talking, I'll look it up.
Yeah, so the next figure, they're looking at how HERV-K could possibly, the expression of HERV-K trigger the innate immune response. So because they see this they observe this HERV-K DNA in the cytoplasm of these senescent HMPCs, which they saw via fish, they think that possibly that this cytoplasmic DNA could be recognized by CGMP-AMP, so CGAS, and trigger the activation of the innate immune system, which would make sense. Like out of nowhere, if I'm becoming senescent and suddenly there are these retroviral-like particles, all these DNA sensors could be activated. So
For obvious reasons, I liked this one too because I spent much of my time thinking about CGAS and DNA sensing. But I really liked sort of the mechanistic tie here because so we get global de-repression. Who cares? Why does that cause senescence? Well, if we specifically de-repress HERV-K, why does that cause senescence? HERV-K is kind of interesting because it can actually reverse transcribe and make DNA to hit the DNA sensors. Other random stuff that got de-repressed doesn't make the DNA to hit DNA sensors. And so this has this unique ability to turn on the innate immune response that had already been linked to SASP.
Yeah. So then they show in figure 4 actually this innate immune activation, and these SASP factors that are associated with the DNA sensing. So I should say the SASP factors. Let me see in B. Yeah, cytoplasmic DNA exactly activates STING, and then REL-A, and then also SASP factors. We can see things like STING-associated factors like IRF3 and REL-A and TBK1 that are increased in the mutant, so the HGPS and the WS, but not in the wild type.
Can you come back to the microphone?
Oh, sorry.
That's
Sorry.
all right.
When you move away, we can't hear you.
Sorry. I was like looking at my paper on my actual paper that I printed. So I'll repeat that. Yeah. So that we see that there are STING-associated factors that are increased in the mutant cells. So things like IRF3 and REL-A, and in the HGPS and in the WS, but not in the wild type. And then via qPCR, they also show the increase in the mutant of these innate, these that are related with the innate immune activation. We can also see inflammatory cytokines like IL-1 beta and IL-6 that are increased quite significantly, especially in the mutants like WS, which was one of the progeroid syndromes, was even more so, almost 20-fold higher with IL-1 beta, which was interesting. And then via ELISA, they also show IL-6. Once again, I don't know why, if anybody thought of like the difference between these two mutant cells, why one of them was so much higher, the IL-6, than the other.
I don't know these diseases at all. This is based solely on figure 1A, which is my sum total of knowledge about these two things. But the drawing makes it look like there's one mutation in the HGPS and there are two mutations in the WS. So I could imagine that because the WS is homozygous for the mutation, that would make it a bit more dramatic. But for example, much of what that mutation is, and I only am guessing about the homozygous heterozygous thing because of the X's in that figure.
Okay. Yeah. I guess anybody else that were to know about these progeroid syndromes specifically could maybe give some insight into that. But either way, it's still interesting that IL-6 and IL-1 beta are so significantly increased compared to the wild type. And then also the inhibition of HERV-K via shRNA decrease the phosphorylation of some of these innate immune factors. And where we can see in H, where are we in H? Because this one also goes all the way to S, FYI,
Yes.
this figure, which are very large. And then we can see that the knockdown of STING, so in addition to this, the blocking downstream effector of HERV-K activation via the knockdown of STING reduced the inflammatory response and the SASP expression, which we see in, I think it's I. Yes, in I. So we see the SH STING versus the SH control. We can see the here. Mine is in black and white here, but I'm pretty sure that it is decreased. Let me just, yeah, exactly. Sorry, because I printed it in black and white. You can see the fold change, and I was like, it says it's decreased, and I'm hoping it is. But yeah, so it's decreased quite significantly. IL-1/6, IL-8, IL-1 beta, etc., which is obviously alleviating the senescent phenotype with the knockdown of this. And then they bring in this drug, abacavir, which Vincent was talking about
It's an
before.
RT
Yeah.
inhibitor. Yeah.
Abacavir.
Yeah, so now you can't make the DNA from the RNA that was being transcribed with the herbs. So we stop
Exactly.
reverse transcription.
Right.
Exactly. So then with this abacavir, so they add abacavir, and then we see that there is a decrease in the mutant cells. We see adding it, there's a decrease in the HERV-K DNA, quite significant. And then we can also see in M, the interferon beta and IL-1 beta and IL-6 is also decreased with the abacavir treatment. So I wonder, abacavir seems like a pretty amazing drug, I have to say. And then we can also see that in M, let me see, from M to O, also alleviated senescence, the senescence-associated phenotypes with the abacavir treatment.
So, as a reverse transcriptase inhibitor, I don't remember abacavir. Is it specifically blocking catalytic site on RT? You know, does it have some kinds of side effects like some of the other?
Probably.
Yeah, I don't know this one other than that it sounds like magic.
Yes.
So yeah, because as Angela said, it sounds like a pretty great drug. I could imagine some people being like, I want to get some of that.
No, don't go browbeat your doctor for abacavir. No.
Okay, yeah, I wonder what sort of adverse effects. I'm sure if Daniel
I'm
was here, he could tell
that-
us.
I'm sure it is not
A
completely benign.
life-threatening allergic reaction.
Should ask.
That
Oh.
doesn't sound great.
Well, so many of the RT inhibitors do have some pretty bad.
Yeah.
Should ask Kevin Bacon. He knows.
Yeah,
the
and that's
answer.
this is not a clinical trial paper. They're using this because they've got a retrovirus and they want to shut it down, and they're testing it out in various model systems.
I mean, it is used in people. It's an HIV/AIDS drug.
Right.
It's interesting, Brianna, it's contraindicated in certain HLA types.
Interesting.
Yeah.
Yeah. But I'm not sure it's going to be an anti-old drug anytime
No.
soon.
No,
Anti-old.
I don't think this is going to be the fountain of youth.
So when you use
I'm
this
certainly
drug,
not going to take it.
you use this drug, you decrease the senescence phenotypes that they're looking at. So,
Yeah.
and
Yeah.
you decrease the innate responses. So that's somehow involved, at least partially, right? May not be the whole thing, but innate responses are part of aging. Yeah. Inflammation. It makes sense, Brianne, right?
Yeah, it does. There's actually this whole field with CGAS and Sting and looking at these and SASP that's
Inflammaging.
already been going on. Yeah, it's referred to as inflammaging.
Yeah. Right.
Inflammation.
Yeah, so then in the last end of this figure, in figure 4, from P to S, the last section, they do activation of HERV-K via CRISPR or via 5-ASA, again, which they see that increases the innate immune response. And there's upregulation of SASP cytokines in the young wild-type HMPC. So even in young cells, we can see that they can induce this senescent phenotype as well. And then we have another, so figure 5, figure 5 then has, once again, up until panel S, but here they're looking at extracellular HERV-K retrovirus-like particles. So apart from, they're looking outside of the cell, is what are these retroviral-like particles doing? Are they interacting with neighboring cells? Could they induce senescence in neighboring cells? So they show that, I think this is where they used the conditioned media. Let me see. They did, they asked whether these HERV-K particles produce senescent cells that could be released extracellularly and then convey these signals to non-senescent cells. So they do PCR to detect, PCR to detect the HERV-K RNA in the conditioned media. So I'm guessing these are just the cells that are in, they just take the liquid from the media that the senescent cells are in. And then they take this conditioned media, and I believe this is where they add the conditioned media to the young cells. And then they also see, so they take the mutants plus conditioned media, the wild types plus conditioned media, and they see that there's increase in the HERV-K RNA just with the conditioned media adding it to the senescent cells. Not even the actual cellular components themselves, but just the liquid. And then they also see that via ELISA, there's an increase in the mutant cells, HERV-K, ENV protein, when they add like those that are in the conditioned media. And then also in D, interestingly, they can see the RLVP, so the retroviral-like particles. They can actually see them budding from senescent cells, which I think this is also electron microscopy, no?
Yep.
Yeah.
In D, exactly. So you can see the wild type where it's not evident at all. And then in the two mutants, we can actually see lots of these viral-like particles budding, which I think is really nice. And they can also visualize, just kind of summarizing some of the other panels, because if not, we're going to be here for a very long time.
Yeah.
They do observe extracellular RNA. So after that, then what they do is they take, what is this? The senescent. So the senescent conditioned media, the SCCM, which is the, what do they call it? Yeah, senescent cell conditioned media. And they can see by adding that to different cells, so the young cells and the mutant cells, that there's an increase of these extracellular RVLPs. And the young cells that are incubated with this conditioned media also increase different SASP proteins, different aging-related proteins like the beta galactosidase. What was it, Brian?
Beta galactosidase.
Beta galactosidase, exactly, and other SASP related proteins. They also see this budding. So from those cells that are incubated with the conditioned media, they can also see this viral, like the particles budding from these younger cells, which I think is also insane, right? Because you think that the old cells would only do this, but the fact that a young cell, just by adding conditioned media from the senescent cells, are doing this is mind-blowing, actually.
Yeah,
Aging is contagious.
Yeah,
Yes.
Literally.
This is such an important figure, and in the places where there was some controversy over this paper, I think that this is the one where people were like, "Yeah, but this figure
But
kind
this
of."
figure, yeah.
But this figure gets rid of some of the criticisms, because basically, if only a small number of cells were having this de-repression happen, but those cells actually made infectious virus, that infectious virus could go infect the young cells and sort of make them senescent as well, and that can go on from cell to cell. So you don't actually need a huge number of cells to be de-repressed in the first place, because actual viral particles are being produced that can take a senescent cell, can kind of turn its neighbor into a senescent cell.
But even
Which I
if
guess
you,
by a lot, yeah. Sorry, Vincent.
even if you have an objection, you in a few paragraphs they actually produce particles from vectors
Right.
in the absence of cells, and they infect those. They use those to infect, and you get the same aging phenotype.
It's like Koch's postulates for
Yes.
aging.
Exactly.
Yeah.
Right?
Yeah,
And
this was the figure that sort of got rid of some criticisms.
That is blocked by abacavir. That turns on the sting pathway and causes all these cytokines to be made. And if you use an antibody against the envelope, you block it. So all of and these are all experiments you don't have to cover now, Angela, because I just mentioned you have to go over the figures.
We won't
All
go to
of
S.
this really wraps it all together, which says that these viruses are doing it. They're inducing aging and senescence markers and inflammation. It's really pretty cool.
Which also makes me wonder, which is maybe, I'm just thinking, like when we get blood transfusions or when we get any sort of transfusion or any sort of tissue, like, well, mostly it would only be blood, but I mean, all of those senescent cells, let's say I'm 60 years old and I'm giving my blood to someone that's 20 years old. Could I be prematurely activating pathways of senescence?
There are experimental
transitorily?
results. Yeah, so there are results that have been coming out over the past several years where if you take blood from a young animal, transfuse it to an old animal, you rejuvenate the old animal in ways that are consistent with kind of reversing aging.
Why would that work, though? Why would that work?
That's the question, right? How does that work? I mean,
Vice versa,
besides the
yeah.
vampirism headlines that this generates, you got to wonder what's the factor that's doing this, and can we bottle that? And I think this paper is getting at a potential mechanism where you might be maybe in the younger blood there are factors that are repressing HERVs, and you're transferring those. And so it's not the blood, really, it's the repression of the HERVs that are being reactivated in the aging.
Yeah. I
System.
started spending way too much time thinking about pigs
Yes.
at this point.
Yes.
Because as Vincent pointed out, people have been thinking a lot about pigs for xenotransplantation, and there are those pigs that have had all of their
They've had all their PERVs
HERVs
removed.
Yeah. Yeah.
And of course, those pigs are being generated for transplantation purposes, and so they're probably not staying around to be elderly pigs.
Most pigs don't when they're kept by humans.
Right, but now I want to know what happens if the PERV-less pigs get elderly.
True.
Yes.
I think the pigs are not terribly healthy.
The PERV-less
true.
pigs, right. Well, in the process of removing the PERVs from the pigs, you have to do a whole lot of stuff that, I mean...
But I also think the PERVs may be providing essential functions that we don't know about,
Right.
most likely. So it's not straightforward, just take them all out, right? And it's
No.
not straightforward just to give you reverse transcriptase inhibitors to prevent you from aging, because that's going to interfere with other elements that you need. So, yeah, there's not a aging cure around the corner, but it's very interesting.
Then monoclonals against this HERV-K are not necessarily going to do the job either. But this is a very interesting mechanistic insight.
Oh, but the experiment in mice, let's skip to that, Angela.
Yeah, they do
what do they do?
a
Tell us about that.
monoclonal. Let's see, the mice. I think the mice is the last figure. Figure
so
7.
we skipped the primates, but
The
tell us
primates, which they show the same. The primates basically they just show that it also different biopsies from different tissues in primates, and then they put them in culture, and then they see also the same phenotypes, the same senescent phenotypes, which is interesting. Like, so it's maintained in primates.
So they also have some human tissues
and humans,
where they see this HERV-K is higher with age, and serum from old people increases HERV abundance. When you add them
There's
to
another aspect to this that isn't mentioned in the paper at all, and that is that on a calorically restricted diet, animals live longer.
That's right.
All of them, including humans.
Yeah.
And so how does that fit into this picture?
I don't know.
It's a good question.
It may be. I mean, senescence is not just PERVs or
it can't be.
HERVs,
It
right?
can't be. It can't be.
Or ERVs. It's got to be other things as well. So.
I wonder what a
They
bristle...
also. Sorry.
I was going to ask what a bristlecone pine tree's herbs look like. It's the
'Cause,
longest-lived organism on Earth.
The bristlecone
Bristlecone
Bristlecone pine.
Huh.
Yeah, it's over 5,000 years old.
I'm sure. I don't think they have endogenous retro, but they may
Or
have
for
other.
a vertebrate, how about Greenland sharks?
Oh, yeah.
All right. Sorry,
lived for centuries.
Angela, tell us the mouse
Angela.
experiment.
I was just amused by the cartoons of the young and old
The young and old, yes.
primates and humans.
So
Yes.
cute. So cute. And also the fact that I'm sure it's the elbow that's up behind them, but it kind of looks like Quasimodo because they didn't do
Yes.
like an elbow hole. So it just looks
No,
like they have
right.
terrible, like, what is it, syphosis? But it's, that was funny. I laughed at that. But, and the faces of the primates, go to figure 6 if you can to look at the little old monkey. Yeah. Very cute. So after we jump over that, we see that targeting endogenous retroviruses alleviates tissue aging also in mice. So here they have a different endogenous retrovirus that they call, it's the MMTV, so the mouse mammary tumor virus.
Yep.
Exactly, which is basically similar to that we have in humans, to the HERV-K. It's a HERV-K counterpart, I guess, in mice we can call it. And so then they show that in...
It's a MERV.
That's right.
MERV kind of sounds like MERS, and then it makes me
was
think
on
of
TV, wasn't
coronaviruses.
Merv Griffin.
That's right.
Yep.
Different generation now.
Merv Griffin.
And they see that this MMTV, or I don't know how to make that any shorter, but in old versus young mice, that there's a very increased signal of the DNA in the old mice. So once again, they're comparing, just like we did in other species. They can see that in mice, it's also like that. And then they also do, intra-articular injections with a lentiviral CRISPR construct. And they see that, well, how many days was this?
That's
This
going
is
to
an
repress the
old-
MMTV, right?
Exactly, repressing
Right.
the virus. And they see that these are 21-month-old mice. So these are considered like senescent elderly mice. These are live mice. We're no longer in cell lines. These are actual mice. So they see after, 10 weeks that there is alleviation of so this was very interesting. So they see that their grip strength gets better. So by repressing this endogenous retrovirus in these elderly mice, so these are mice that are already old, that have these retroviruses, the MMTV, but that by repressing this virus, they see that they get better grip strength. What functional movement is improved in the mice. It increases their cartilage thickness, and there's just joints in general. They have better mobility, and overall, it says improvements in tissue aging. But I'm guessing that's just everything I just said, which I think that's so in vivo. They're actually seeing that just by repressing this virus, they see this change in the senescence phenotype, which is really, really cool.
I'm reading about how you test the grip strength of mice.
Yes.
That seems really fascinating.
It's kind of cute, actually.
yeah, it is.
Is it like on a toothpick or something?
No, so it looks like what
Like-
they do is they put them on a grid and pull the grid and wait to see how much force it takes for them to.
That's
to
how we used
stop
to
grabbing.
To pull the rug out from under the mice.
That's
yeah.
how we used to tell if mice were paralyzed with polio virus. They can't grip
Oh,
the
really?
cage anymore, and they let go. Yeah, an early sign before the limb actually gets paralyzed, they can't grip. So, Dixon, how's
Oh,
your
wow.
grip strength?
It's very good, by the way, because I use walking sticks in order to move, and my hand strength is
see.
very, very good, except for the arthritis, which I,
Because we could give you this lentivirus,
No,
or
don't do that,
Or
don't do that.
abacavir apparently does the same thing in mice, right?
Exactly. So then they actually give abacavir to mice. I'm sure this has never been used in humans, but they give it to the mice.
No, it has been.
And
It's
they
given
see
for
that-
AIDS.
It's
It
it's
is.
given as,
okay,
a,
no,
as an
it's
HIV
true. It's a
drug in
retroviral
humans.
therapy.
Yeah.
It's true. But I mean, it wouldn't be given for aging
No.
as of now.
No,
It would be
no.
given in a pathological context. Yeah.
Hey, Bill, though, that's a good question. Could you use it in an off-label thing because it's approved for HIV/AIDS?
You
Could a doctor?
could, but
We shouldn't
don't.
plant that
okay?
seed.
Just don't.
No, we shouldn't
no,
plant that
please
seed.
do not do that.
How would you do it? Would you have to use it every day for the rest of your life? Would you need to start when you were young?
Well, I
You-
don't know. That's a
Yeah.
good question. I'm just wondering
Well, I mean,
if
yeah. So they're seeing, I mean, in here, they're seeing a reversal to some extent of one symptom
Yes.
of aging in mice. And I really don't think that that's worth the downsides of.
so. They gave it to them for 6 months in their drinking water,
Yeah,
which
exactly.
is a
And
good
they
fraction
saw-
of their life, right?
it's-
Yeah.
Yeah.
And they
So-
saw bone density increase, which I thought was interesting. So they saw that by giving this abacavir to the mice, like Vincent said, in water, they see bone density increase. They see the thickness of the cartilage also increases around their joints. They see less cartilage degeneration because it's thickening. What else do they see? I already said the thickness of the cartilage. Yes, they see IL-1 beta is decreased, which was associated with an innate immune response and inflammation and senescence. They see that is also decreased post this abacavir treatment. Grip strength also increases. So basically everything that we were talking about before, they see just post abacavir treatment for 6 months, which I think is really interesting, actually. Obviously, in humans, we don't know what all
I
of this
could
could mean,
see-
but
Someone designing a clinical trial with an RT inhibitor to do some very careful observations on some of these measurable senescence phenotypes, right?
yeah.
Like osteoarthritis.
Tell me about it.
Or also
most
maybe
common-
I'll
joint-
take it.
You would enroll in the trial for that?
Well, if it would have some effect on my osteoarthritis, I sure would.
So it says
because
it's the most common joint pathology with aging.
it is.
Huh.
Absolutely.
Abacavir side effects. Commonly reported side effects of abacavir include arthralgia, cough, fatigue,
right.
lethargy, myalgia, pruritus, vomiting, chills, and malaise. Other
take
side
some.
effects include hypersensitivity condition, pharyngitis, and tachypnea, which that's got to be a breathing
increased
problem, right?
breathing. Yeah, increased breathing.
Yeah.
Oh, you're a vet. You should know that, right?
But I feel like maybe we could do some sort of, because it's used in a pathological context, like with people that have HIV, I don't know if it's only used during AIDS treatment, but perhaps there could be like a joint clinical trial for those that are taking abacavir, just looking at their bone density, looking at their grip
Yeah.
strength, things like that, because they're already on the treatment.
Yeah.
Maybe they could have,
Yeah, that's right.
maybe we
you
could also
do
look
from
at
medical records case control analysis or something.
Exactly. That would
Yeah.
be interesting.
I mean, it's not
And that,
perfect because it's not an RCT,
of course not.
right? You don't.
Right.
Yeah.
Well, right, but you could find, I don't know how commonly abacavir is given for HIV, but there might be people who are on it and people who aren't, who both are being treated with antiretrovirals, and you could compare. Of course, if they're both being treated with antiretrovirals, it's possible that other antiretrovirals would inhibit
Yeah.
HERVs.
yeah, then they
and
could
so
look
you
at
might,
others.
You might have
Yeah.
a confounding factor there.
Well, all
So-
the people who are on antiretrovirals for AIDS, you could look at them and see how they're
yeah,
doing
exactly. Right.
with
Exactly.
osteoarthritis, right? But I don't know if it inhibits HERVs RT. That's the thing.
Yeah,
We'd have to
so
look.
it looks like abacavir is often given in specifically in combinations, and it's a nucleoside analog, specifically a guanine analog. So I don't know if I'm feeling like a guanine analog should go into all the people.
No.
Yeah, probably not.
No. This is not the treatment for aging, but
Maybe we
this
could-
paper is showing us mechanisms
Yes.
that we
Right.
would like to know more about in order to try and target them maybe a little more
I mean,
specifically.
maybe we could develop a protease inhibitor for the HERV-K, right, which is needed for particle formation, and that would be less toxic. Who knows,
Yeah.
right? But what I would like to know is if these endogenous retroviruses are so, I mean, they make you age, why haven't they left the genome over hundreds of thousands to millions of years, right?
because this is a post-reproductive
Yeah, it must be
selection.
it.
Yeah.
Yeah.
Right.
So things related to aging, I mean, once you've reproduced,
true.
evolution really doesn't care what happens to you.
See,
So
nature favors the young.
yes.
Sorry, Dixon.
It's okay.
Me too.
I've had my
Well,
day.
sorry, all of us, right? Because we're all headed there eventually if we're lucky. Yeah. So this is
That's true. Post-reproductive. That's absolutely.
there's just not
it,
enough
yeah.
pressure against it, and we're over evolutionary history. We're constantly getting every several million years. We're getting more endogenous retroviruses or more retroviruses that are endogenizing in
Right.
various species.
Plus, some of them are doing beneficial things,
Yeah.
and in
Yes.
fact, beneficial things that are specifically
Sure.
helping with reproductive success.
Yep.
Yes.
If we think about this incisions, so maybe HERV-K has a positive effect in some aspect, but also this
Well,
negative
young.
effect later on.
Yeah,
Right,
when
so-
you're young, and then, but old people, you've already reproduced, as Alan said, it doesn't matter. There's no selection against it.
Yeah.
Yeah. So that's why they're there. Very interesting. I think it's a cool paper.
Yeah.
I think the
Very cool
finding
paper.
of the old cells literally taking the conditioned media and adding it to the young cells blew my mind that it
Yeah.
could induce that. That's also terrifying. But
I mean, it's the
Somehow-
particles. It's the particles,
particles,
in the media
exactly.
that's doing it, right?
Aging
Of course,
is
of course, of
infectious.
Aging is
Yeah.
infectious. Yes.
Yeah.
Apparently, aging
of course,
is infectious.
literally though.
But the
And to
idea
have
of
that,
giving blood. What if
Ah,
old people give blood, right, Dixon?
They do.
of course.
And so you give it to a young person. Are they going to age quicker?
That's what I was thinking before. That was my
Yeah, that's
question. But I
Angela
don't
brought
know.
that up, and
I don't know how. If I
How
don't
would you
know.
know unless you
I don't
it's
know.
not
The-
enough.
the tr-
it's
yeah,
not.
that's what
It's
the
going to be
question is: the
right.
transients and
So
the
first of all,
levels.
Yeah.
first of all, you're
Oh.
giving, if you're giving HERVs, all people already have them. So
Yeah.
you get your, some 75-year-old donated blood, and you get a transfusion, and you're 25 years old. You temporarily have maybe some extra HERV particles, but your body's already got this same virus in it. It's going to wash out. And also, if you're young and getting a blood transfusion, it's probably because you didn't have a great day in the first place.
Yeah.
And the effects of
Unless
that
you were in
are-
an accident, if you had like
True.
a freak
Yeah.
accident. and
that's
then
what I
you-
mean. You probably something happened that you needed a blood transfusion. And so the effect, whatever effect these have on causing some senescence in a few cells for the days or weeks that they're actually in you, are not going to be noticed against the background of healing from your injuries in the crash.
True, it would
There's
be
a-
transitory unless you
Yeah.
had.
Yeah. It's too
Mm-hmm.
it's too
Too transient.
Transient.
Yeah.
That's the word. Yes.
Transient,
Because you have
yes,
to give the
that's the
mice
word.
for a long time to get it.
Well, you can also do
Yeah.
parabiotics experiments here. You know, you can take an old rat and a young rat and link them together by their
Oh,
abdominal tissues. And then does the young rat get older or does the old rat get younger?
Yeah, I mean, I think that that's probably a more interesting way to think about it, because I was just looking, and they, when they looked at the in vivo stuff, they're looking at tissues for the most part. And so the other question is, how much of are these particles actually in blood?
Right.
right.
So to summarize,
But they did
this,
take serum. Sorry.
Yeah,
they
they
did take serum from the elderly, and they
yeah.
saw
Yes,
from the
yes.
serum.
Yep.
Sounds
that
like
there
we're
was
writing a grant.
HER2.
so to summarize, we think from this paper that these endogenous retroviruses are somehow inducing senescence phenotypes, and it seems that the innate immune response and inflammation is part of that, and it can
Yes.
be passed, and the virus can go from cell to cell as viruses are wont to do, and that can cause senescence in another cell in another organism. Very interesting. So yes, aging is infectious, isn't it?
It is.
But I don't think
Maybe
in the
that
real
should-
world that because I think the titers of these herbs and people are very low, so I doubt you would pass it on to anyone. You know,
I don't.
And
Well, the
even,
Unless
titer
you were
was-
in a parabiosis
yeah,
situation.
and even
Yeah.
a blood transfusion is too transient, so I don't think it's a problem.
I also wanted to add, I forgot to say in the beginning, because I also read this, that yesterday actually came out in Cell. There was a preview on this paper called "Is Aging a Retrospective Event?" And retro was in
Yes.
quotations, which I thought that was cute, by Nirmalya Dasgupta and Peter Adams from the Sanford Burnham Prebys Medical Discovery Institute in La Jolla, California, which was also in Cell yesterday, a preview on this. And it was also really good. If you want like a digested 1 page and a half that explains it all, it's really good.
If you want us to digest it, is that what you're saying?
I mean, I made it quite complex. It's not my field. I hope it was.
Okay, can you put the link to that in the show notes?
Yes, for sure.
I will put them in the the actual show notes on the website. All right, thank you, Angela. All right, let's go right to some picks. Dixon, where are we?
We are still with the jazz program, still with the jazz project, and we've covered the saxophones from alto to tenor, and now we have the ultimate baritone saxophone player, Jerry Mulligan. And Jerry Mulligan played with a number of West Coast musicians, was married to Sandy Dennis, by the way, was a remarkable saxophone player, just absolutely wonderful. Played a lot of very tender, slow, moody songs he became famous for, but his signature song is called Bernie's Tune, and Bernie's Tune is a very fast-paced, I think it's a saxophone quartet that actually plays it, and it's wonderful to listen to, and I highly recommend it. He was just a fantastic musician. I saw him a lot of times at Birdland, and I really did enjoy his music quite a bit. So he's I don't have a second pick for baritone sax. This is the guy that you got to go. If you want to know anything about the baritone sax, this is your player.
So this is a really deep saxophone, is that right?
It's big. It's very large.
Yeah.
It's almost as big as Jerry Mulligan. When you see videos of him playing, the instrument is quite heavy and quite large.
Well,
But it's great.
I've heard of the name, but I don't know any of his music, so I'll have to check it out.
Yeah, absolutely. Check it out. I don't think you'll be disappointed.
What is the next instrument on your list?
I did harmonica. I think
You did a harmonica already?
I think I did Toots Stillman. I think I did. Yes, I think I did. Oh, look. I it's at home. I'm not at home right now. I'm at the studio. So all of my picks are part of a larger document that I can access. I'll send it to you.
I'm looking for TWiV and harmonica.
No?
I
Toots Stillman? Maybe I'll make that to my next one then if I haven't.
don't think you've picked the harmonica.
Okay,
yet.
harmonica. Harmonica is
All right.
next.
Angela, what do you have for us?
Okay, so I have the scientists discovered the world's largest animal. So this is technically even larger than a blue whale. It's not a mammal, but it's an animal, and it is a siphonophore. So siphon. I believe that's how you say it. Siphonophore.
Right. So this is the
That's right.
longest
That's
animal,
cheating. That's
but it's
cheating.
very
Longest
That's
thin.
cheating.
animal. Longest
cheating.
animal. It's 150 feet long.
Yeah.
So for those of you that are in meters, I think that's what, like 40, 50.
Probably
It
about
was like
50
50 meters. Yeah, because it's like
Yeah.
3 feet per meter. So like 50 meters long. So wait, is that bigger than a blue whale? Now I'm
Well, it's-
questioning myself.
Oh, length of a blue whale. Vincent's got the Google that.
Let's see. How long is a blue whale?
Okay, no, 25 meters.
30 meters, maximum
30 meters,
length.
yes. Yeah, so it is longer. Okay.
Longer than a blue
good.
whale.
Longer than a blue whale, exactly. So I was.
But
Not
could probably
heavier,
be
not
eaten by a
heavier
blue
than-
whale.
No,
which
just
is a much,
long.
much larger
Oh,
volume.
definitely. So these, so siphonophore, you can look it up on Wikipedia or on Google. They're really cool. They're actually from the order Hydrozoa. So they're basically, they are considered an individual organism, but they're composed of all of these, I think they call them like medusoid, kind of like
You
jellyfish-like
string a bunch of jellyfish
polyps.
together and you get
Exactly.
one of these things. Yeah.
Exactly, and they're all from like one fertilized egg, but they're considered one organism, and some of them have bioluminescence, and I've actually seen them before diving, but obviously never one that was 150 feet long. But they're really cool. They sometimes form like tubes, and some of them can cause like urticaria, so you don't want to touch them because they can make you
The nematocysts
break
will
out.
get you.
Exactly, yeah. But they're really, really fascinating, and I thought that was cool that we found the largest one.
So they're
yet,
carnivores, right? They're carnivorous.
Yes. Yes,
What
which
do
is
they eat?
also
They eat minnows and the small fish
other fish. Yeah, other fish.
that get tangled up in their tentacles.
Wow. How about that? I didn't know this.
And then they slowly digest them. It takes a lot, which is also
Exactly.
slightly horrifying if you see it, like
Yes.
slowly digesting a fish, because they're quasi-translucent. So yeah,
That's right. That's right.
And where do they? Which oceans are they living in?
All over.
All over.
I believe they're everywhere. Yeah, when
They're
I lived in the
abyssal
Canary
organisms,
Islands.
aren't they?
This
Abyssal?
one was found in Australia,
They're found
Yeah,
in
this one's
deep
Aust-
waters, so I think.
Yeah, they're
where
pelagic.
frightening and weird creatures
they're
are
deep
often
water,
found.
so you're not going to find these swimming on the coast.
No,
right?
but they might migrate to the surface during the nighttime. There's a lot of organisms that go up and down.
Cool.
Very
I've seen siphonophores, smaller ones near
right.
the coast when I've been diving. Not obviously to this extreme, but I've seen smaller ones diving before.
How thick are they? About a finger's thickness?
No, it depends. Some of them can be like
6 inches.
like even 6, 10, 15 inches across, and some of them are really thin. I think there's, what was it? I read it on Wikipedia, 175 species. So they all vary in size and length. Obviously, this is the
So
largest one, but they're not all that
they don't.
big.
There's not a mouth at one end because
Oh,
each
no.
each is a.
They bring it into the center part of the medusa, and then they wrap up their stomach like organelle around it and secrete digestive enzymes, and that's
Is
how
this
they-
like a tapeworm segmented?
No, not like tapeworms have no
Well,
mouths.
yeah, siphonophore,
No, it's not that- it's not
siphonophore
that.
kind of is segmented, right? Because it's all the individuals' pieces joined
I
together.
guess you could see it as segmented. Yeah, that wouldn't be wrong.
But it's not quite
I'll give you a
segmented
B+
in the same
on
way
that
as a
one,
tapeworm. What?
So it's not going to wrap itself
No,
around you,
no,
and
no, it
it's
doesn't
like
do
a
it that
snake.
way.
It's not going to do
No,
that.
No,
it's
the
not a
tentacles
No,
are
no, no.
sticky, and then they contain nematocysts
That's
that
very cool.
stun their organisms. With,
They're tiny fish that they eat. They're not grabbing like tuna or anything. They're grabbing
Right.
a little
Like,
tiny
you know,
fish that gets stuck to it.
a Portuguese man of war.
All right, and I have one last question. How long do they live?
Probably
no
forever.
So they must not have endogenous retroviruses.
Right.
Actually,
probably do.
I'm sure
probably
they
do.
Yeast have endogenous retroviruses, so yeah.
Yeah, yeast do. So do I'm not going to do a search. As
They've
G-
been around for 640 million years, but I can't see how old they can. How old?
Siphonophores are yeah. That's an old phylum. It's very old, I think.
Yeah. They look it, too.
How old can they be?
They're very primordial looking organisms.
That's
Yeah.
right. That's right. Yeah, they're almost like 99% water.
Yeah.
All right, very good. Brianne, what do you have for us?
So I have an article about a comet, specifically a particular green comet named 2002 E3 ZTF, a very descriptive name, that is getting ready to pass by Earth for the first time in 50,000 years, and that will be visible from Earth. It will be at its closest on February 2nd, but due to the new moon this weekend, a lot of the idea is that it might be most visible this weekend, even though the second is when it will be closest. It will be near the Big Dipper. And it will, depending on how you see it, you may actually be able to tell that it's sort of a green, smudgy comet. It has a unique sort of form of carbon, which is why it is green. It's a type of dicarbon. And because it has sort of been such a long time since it's been here, and it's really unclear when it might come back or kind of what's going to happen to it afterwards, a lot of astronomers are spending a lot of time trying to get as much data as they can in this pass. So this article was the first I had learned about it, and now I'm going to try to make sure I see it sometime soon.
That's pretty cool.
Because if you don't, you'll have to wait another 50,000 years
Exactly.
for it to come back.
Well, you don't know that. You don't know.
With our retroviruses, I don't think you'll make
Better
it that
suppress
long.
those herbs. Yeah.
Alan, what do you have for us?
I have the second in my video game picks. So I'm picking odd indie video games that people might not have heard of, but that I think have really compelling stories. This one is Golf Club Wasteland, which it's available on most platforms, I think, desktops at least. And the game itself is fun. It's a golf simulator where you pick the angle and the force that you're going to hit the ball with, and there's a hole somewhere else on the screen that you need to get it to in as few hits as possible. But you play as this little guy who's in a spacesuit with a jetpack, even though he's on Earth. And it's a post-apocalyptic landscape of Earth with buildings that are run down and breaking and various
Wally World.
pools of toxic stuff sitting around. And as he plays golf across this abandoned, destroyed Earth, he's listening to a podcast.
TWiV?
Which you quickly learn it is not TWiV. It's a podcast that's being beamed from Mars, where the surviving humans, small population of surviving humans, have set up shop. And this is, in fact, a touching story behind it that he's experiencing along the way and that you uncover as you shoot these various holes, and also a social satire of something called the mindset, which is this view among certain Silicon Valley folks who are a little too high on their own hubris that the Earth, we're not going to be able to save it. The environmental damage is too bad. And what we really need to do is colonize other planets. And so that's the driving force behind a lot of these things like SpaceX and Mars colonization. And you hear a tremendous amount of hype about that. And it is driven to a large extent by very wealthy people or influential people who are stuck in the mindset that we're somehow going to be able to escape Earth and not have to deal with all the problems that they've helped create. And this is a rather dark send-up of that, but couched in terms of a fun little golf game you can play.
Looks nice. The graphics
Highly recommend it. The graphics are really cool. It's a neat design, and just an interesting story that plays out mostly in this podcast the guy's listening to.
Too bad it's not TWiV.
It's not TWiV, but if it were, it wouldn't get the point across.
That's right.
True.
All right. Cool. Well, I think Brienne is the only one who's probably going to check that out, right?
Well, maybe Angela.
I might.
Right?
Angela, do you game?
Not so much. I used to. My brothers are all huge gamers, but
Okay.
I like the color palette of this. I like the teal and the
and
blue.
it's a low. It doesn't require much of a computer to run. So it's on Steam. It's ostensibly a Windows game, but most of the games on Steam now run on other platforms because they've got a compatibility layer.
I used to play a lot of Super Smash Brothers. Yes, those were my kind of games.
Okay, we're
Age
big
of
Nintendo
Empires.
fans in this household.
Oh, yes.
I'm not so much into Smash, but I think my daughter was for a little while.
My pick is a commentary or perspective, I should say, by Paul Offit in the New England Journal of Medicine. It's called Bivalent COVID-19 Vaccines, a Cautionary Tale. And he sent this to me last week. He said he wrote it because his last discussion with us on TWiV inspired it. It's basically a summary of what he said on TWiV. He goes through the history of COVID vaccines, and it talks about the bivalent vaccine that were initially tested, what results we got from them, and how, he says there's no evidence that they're any better than the ancestral and that they're going to provide any additional protection. But the last paragraph, he writes, what lessons can be learned from our experience with bivalent vaccines? He says, fortunately, variants have not evolved to resist protection against severe disease that's offered by vaccination or infection. If that happens, we will have to create a variant-specific vaccine, although boosting with a bivalent vaccine is likely to have a similar effect as boosting with a monovalent vaccine. Booster dosing is probably best reserved for the people most likely to need protection against severe disease, specifically older adults, people with multiple existing conditions that put them at high risk, and those who are immunocompromised. In the meantime, I believe we should stop trying to prevent all symptomatic infections in healthy young people by boosting them with vaccines containing mRNA from strains that might disappear a few months later, which I think is a very nice conclusion. So it's open access, so you can go check it out. And then we have a listener pick from Lisa. This past weekend, we were visiting Big Bend. And when visiting with my husband's friend who lived in the area, the subject of canoeing or kayaking down the Rio Grande came up. And that reminded me of this great documentary, The River and the Wall, that I saw a few years ago and that she had also seen. It provides a link to that. I was so glad I saw it because it enabled me to see parts of the Rio Grande that there is zero chance I would ever make the physical effort to get to, but that are well worth seeing. Despite the title, this is much less political documentary than you might expect. I'm pretty sure you could watch it with a viewpoint diametrically opposed to mine on the border wall and still find it valuable. One very disappointed virus note on the trip, a different friend of my husband's whom we visited in the area, who is in an age category in which I would have thought any doctor would have encouraged the use of Paxlovid, just weathered COVID-19 with no antivirals. He said his doctor didn't want to put him at risk of Paxlovid rebound.
Oh, brother.
Paxlovid rebound unless he started to get really ill. Oh, my gosh. Unless he started to get really ill, at which point it's too late.
That's right.
There's no Paxlovid rebound. It is COVID rebound. Thanks again for the valuable podcast. Lisa is in Austin, Texas. There's still some people who believe it's Paxlovid rebound. It is not. It is not Paxlovid rebound. There's no reason not to take
Vincent,
Paxlovid.
is it Paxlovid rebound or not?
But of course, you may be taking drugs that are not compatible with one of the components of Paxlovid, in which case you need to take remdesivir. Okay? But Dixon, you took Paxlovid.
And so did you.
And so did I. And it had no rebound.
Two satisfied customers.
But NF2 is not a clinical trial. I
But
understand
if you had
that.
had some kind of rebound, it would not have been the Paxlovid rebound. It would have been COVID rebound.
COVID rebound. It's a natural part of the pathogenesis. So stop trying to prove it's Paxlovid. You're discouraging people from taking Paxlovid, and you're killing them.
But you are right about the nasty taste in your mouth.
It's okay. It's only 5 days, right?
It's only 5 days.
You can deal with it.
I did.
All right. That's TWiV 977. You can find the show notes at microbe.tv/TWiV. You can send questions, comments, your picks of the week to TWiV at microbe.tv. If you enjoy our work, we depend on you to support us so we can do this. We are a nonprofit. We don't do ads. We don't monetize. We are a nonprofit who depends on our wonderful donors like you. So go to microbe.tv/contribute. Any amount that you can contribute, we welcome to help us keep this going. Dixon Despommiers at trichinella.org, thelivingliver.org. Thank you, Dixon. Good to see you here.
It's a pleasure to be a guest in your bailiwick.
My bailiwick.
Your bailiwick.
Alan Dove
Bailiwick.
is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here. I learned a lot.
And Angela Mingarelli is at McGill University. Thank you, Angela.
Thank you. I will add that I am Immune Vet on Twitter. And that, yeah, I had a wonderful day today. I hope you guys learned a lot. I definitely did.
What are you, immune what?
Vet.
Vet.
Oh,
Immune vet.
Vet.
VET, yeah.
Twitter.
Immune vet.
Immune vet. All right. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.